Important information you should know about avian influenza.
Samples for the laboratory investigation of suspected outbreaks of avian influenza can be obtained from sick and recently deceased birds, as well as the environment they inhabit. Oropharyngeal or tracheal and cloacal samples, as well as blood samples, can be obtained from both live and dead birds on site. If it is unfeasible to take a cloacal swab, a fecal sample can serve as an alternative. If you suspect an outbreak has occurred, collect 20 cloacal and 20 oropharyngeal swabs from the flock. A minimum of 5 birds should be collected for necropsy. The organs needed for virus isolation are the brain, spleen, kidney, liver, intestine, and respiratory tract. 20 blood samples should be collected. In all the mentioned sample collections, birds that appear sick should be targeted.
Respiratory specimens should be kept at 4 degrees Celsius for no longer than 3 days. The viability of some pathogens from specimens that are frozen and then thawed is greatly diminished. Samples should be transported with the appropriate packaging and according to instructions given from the laboratory. Specimens must be labeled clearly, stating all the information requested by the health laboratory.
Keep samples cool during transport. Data obtained are more accurate and it reduces bacterial diversity. Specimens should not be exposed to environmental extremes. Blood samples should be stored at 4 degrees Celsius, and specimens should be kept sterile. Keep specimens in a tight container to prevent possible escape of infectious agents or cross-contamination of multiple samples.
Possible lab tests that can be done to diagnose avian influenza include virus isolation, as well as real-time reverse transcriptase polymerase chain reaction assays on pharyngeal swab specimens. Additional characterization can then be done on positive samples, and these include serotyping or genetic sequencing.
In order to determine the morphology of a virus, a sample of the virus needs to be stained. Various staining methods can be used. For example, the shadow casting method, which deposits heavy metals such as platinum or tungsten onto the surface containing the specimen at an oblique angle. This creates a shadow of the specimen and the morphology can therefore be determined. The negative staining method is when a film with the specimen on it then has a drop of negative stain, usually sodium phosphotungsten, added to it. The stain surrounds the specimen and leaves the specimen white with a dark background. Lastly, using a positive staining method, which includes staining the specimen with uranyl acetate, viral components then absorb certain salts, making the virus appear darker than its surrounding. There are 4 basic morphological forms, namely icosahedral, helical, complex, and binal. The avian influenza virus has the helical morphology and is coated by an outer lipid membrane called the envelope.
The avian influenza virus can easily be grown in cell cultures. This is a highly sensitive technique for detecting live avian influenza from clinical specimens. Also known as the gold method, it is 100% accurate but may take 2 to 10 days for results. Most avian viruses grow readily in embryonated eggs, and this is the option of choice for the amplification of viruses from avian hosts.
During the process of avian influenza virus isolation, the sample material is inoculated into the allantoic cavity of a 9 to 11-day-old chicken egg. The eggs are incubated, allowing any virus present to grow.
The symptoms of avian influenza include no coordination, ruffled feathers, difficulty breathing, a loss of appetite, depression, bluish coloring of wattle and comb, edema and swelling of the head, eyelids, combs, wattle, and hocks, watery diarrhea, pinpoint hemorrhages, bloody or watery discharge from the nose or beak, and eggs with soft or deformed shells. However, these symptoms could also indicate other diseases. You need to establish the exact nature of the disease affecting the birds. A veterinarian should take blood samples or take a few birds to a laboratory for further analysis.
Eclipse phase is the time interval between successful cell infection and the start of the virus production. It's a time interval during which the virus infection cannot be recovered. Firstly, the virus enters the cell to parasitize it. It includes the attachment of the virus to the cell membrane. The cell gets penetrated by the virus. Once the virus has entered the cytoplasm, it is uncoated. During the life cycle, within the cell no progeny is found, and the virus is not active. The virus disassembles and component parts are synthesized. The genome of the virus consists out of negative sense, single-stranded segmented RNA, which consists out of 3 segments. The viral entry through the lipid rafts and endocytosis after attachment, and primary transcription is started by the L proteins. Viral proteins accumulate and replicate, then enters the maturation phase.
Influenza A viruses such as avian influenza are unique in their ability to undergo variation. They can undergo both antigenic shift and drift. Antigenic shift is when an antigenically new virus appears about every 10 to 30 years. These are the viruses that cause pandemics. Antigenic drift, on the other hand, is evident between these shifting events. It constitutes minor antigenic changes and is the cause of epidemics.
The virus can be transmitted between different types of birds, both wild and domesticated, either airborne through direct contact with infected poultry or indirectly by coming into contact with surfaces that could be contaminated with the virus, such as the water feeders, living area, and feed. The virus is shed into the environment from an infected bird's saliva, feces, and nasal secretions.
The influenza virus has 2 main mechanisms to escape the immune system of the host. Firstly, they have a high mutation rate. The pathogen can exhibit great changes in some viral gene sequences. Secondly, they have the ability to undergo gene reassortment. All influenza viruses consist of an 8-segmented RNA strand. When invading the host cell, the RNA segments enter the host nucleus and is then copied to form many RNA genomes. But if a cell is infected with 2 disparate influenza viruses, the RNAs of both viruses are transcribed. When new virus vesicles are formed, each segment of the RNA is composed by a combination of both infecting virus genes.